Medicine and health sciences have experienced dramatic advances in recent decades. Among these advances, the use of genes for therapeutic purposes, known as gene therapy, represents a promising tool to cure some of those diseases that conventional drug therapies cannot.
Gene therapy. What is gene therapy? An experimental technique for correcting defective genes that are responsible for disease development. The most common form of gene therapy involves inserting a normal gene to replace an abnormal gene. Other approaches use replacing a mutated gene that causes disease with a healthy copy of the gene, inactivating or knocking out, and mutative gene that is functioning improperly, introducing a new gene into the body to help fight a disease. History and development of gene therapy. In 1960, the concept of gene therapy was introduced. And in 1970, Theodore Friedman and Richard Roblin cite the first attempt to perform gene therapy. And first patient to be treated with gene therapy was a 4-year-old girl, treated at an NIH Clinical Center in 1990. Congenital disease called adenosine deaminase or ADA deficiency, which severely affect immunity and the ability to fight infection. In March 18, 2002, sickle cell treated in mice by process of gene therapy. Sickle cell disease is a disorder that affects blood cells, which use a protein cell's hemoglobin to transport oxygen from the lungs to the rest of the body. In October 3, 2002, gene therapy was used for the treatment for ex-kid, children who has severe combined immunodeficiency. In children, triggers leukemia. In April 28, 2008, the world's first gene therapy for inherited blindness. As a result, the experimental treatment is safe and can improve sight. In March 2009, the use of nanotechnology plus gene therapy yield treatment to torpedo cancer. Future possibilities lie ahead through further insight and knowledge. Gene therapy holds many possibilities. There are types of gene therapy. First is somatic cell gene therapy, and the second one is germline gene therapy. Somatic cell gene therapy is a therapeutic genes transferred into the somatic cells.
Here is an image of a somatic cells.
There are examples of introduction of genes into bone marrow cells, blood cells, skin cells, and etc. Will not be inherited later generations. At present, all research is directed to correct genetic defects in somatic cells. Germline gene therapy is a therapeutic genes transferred into the germ cell. Examples are genes introduced into eggs and sperms. It is the heritable and passed on to later generations. For safety, ethical and technical reason is not being attempted at present.
Germline is essential for reproducing the organism, while somatic cell cannot reproduce the organism.
Embryonic stem cell and gene therapy. Human embryonic stem cells theoretically represent an unlimited supply of normal, differentiated cells to engineer diseased tissue to regain normal function. Embryonic stem cells are pluripotent cells derived from the early embryo. Applications of gene therapy to human embryonic stem cells. The ability to produce genetically modified human embryonic stem cells expand the potential of human embryonic stem cells to correct hereditary disorders and deliver gene products for specific therapies. Strategies are being developed to enhance the safety profile of human embryonic stem cell transplant by incorporating genetic elements into the expression cassettes of gene transfer vectors to allow the use of external stimuli to suppress or eliminate uncontrolled cell growth. Use of gene transfer to control differentiation and growth of human embryonic stem cells. To be useful in clinical applications, genetic modification of human embryonic stem cells may be carried out using multifunctional gene transfer vectors that include a selectable marker, a therapeutic gene, and suicide gene.
According to National Institutes of Health in United States, the potential of human embryonic stem cells for gene therapy is the topic. Following derivation, human embryonic stem cells are easily accessible for controlled and specific genetic manipulation. When this facility is combined with the rapid growth, remarkable stability, and ability to mature in vitro into multiple cell types of the body, human embryonic stem cells are attractive potential tools for gene therapy. Thus, there are some cases that the stem cell-based gene therapy is facing, despite of the scientific results with genetically modified stem cells, there are some major problems remaining to overcome. Because the more specific the genetic modification is, the longer the stem cells have to remain in vitro. Even though scientific said that it helps the human with their body, it might harm their body too. Now, let's discuss the types of somatic gene therapy.
Ex vivo and in vivo. Ex vivo is cells are modified outside the body and then transplanted back in again. Called ex vivo because the cells are treated outside the body. In vivo, genes are changed in cells when the cells are still in the body. Called in vivo because the gene is transferred to cells inside the patient's body.
How are the scientists process the ex vivo gene therapy? First, they isolate the cells with genetic defect from a patient. The cells are grown in large numbers in tissue culture plates. Then they introduce therapeutic genes, and they select the genetically corrected cells and it grows. Lastly, they transplant the modified cells to the patient, which means the cells are then returned to the patient to replace the function lost due to inheritance of mutant genes.
In vivo gene therapy. Direct delivery of therapeutic gene into target cell into patient's body. They carried out by viral or non-viral vector system. It can be the only possible option in patients where individual cells cannot be cultured in vitro in sufficient numbers. Example is brain cells. In vivo gene transfer is necessary when cultured cells cannot be reimplanted in patients effectively. Vectors in gene therapy. To transfer the desired gene into a target cells, a carrier is required. Such vehicles of gene delivery were known as vectors. There are two main classes, viral vectors and non-viral vectors. Viral vectors. Retrovirus vector system. The recombinant retroviruses have the ability to integrate into the host genome in a stable fashion.
Can carry a DNA of size less than 3.4 kilobases.
Replication defective virus particles, and the target cells is dividing. Adenovirus vector system. Adenovirus with a DNA genome or a good vectors. Target, non-dividing human cells. Sample is common cold adenovirus. Viral vectors. Adeno-associated virus vector. It is a single-stranded, non-pathogenic small DNA virus. Herpes simplex virus vector. Viruses which have natural tendency to infect a particular type of cell. They infect and persist in nervous cells. Non-viral vector system. Pure DNA construct is a direct introduction of pure DNA construct into target tissue. Efficiency of DNA uptake by cells and expression rather low. Consequently, large quantities of DNA have to be injected periodically. Lipoplexes, lipid DNA complexes. DNA construct surrounded by artificial lipid layer. Most of it gets degraded by lysosomes. Non-viral vectors, DNA molecular conjugates. Commonly used synthetic conjugate is polylysine compound to specific target cell receptor. Therapeutic DNA is then made to combine with a conjugate to form a complex. It avoids lysosomal breakdowns of DNA. Human artificial chromosomes can carry a large DNA. Examples are with one or more therapeutic genes with regulatory elements. There are some disadvantages of gene therapy. These are: long-lasting therapy is not achieved by gene therapy due to rapid dividing of cells. Benefits of gene therapy is short-lived. Immune response to the transferred gene stimulates a potential risk to gene therapy. Viruses used as vectors for gene transfer may cause toxicity, immune responses, and inflammatory reactions in the host. And also, disorders caused by defects in multiple genes cannot be treated effectively using gene therapy.
Having a disadvantages, gene therapy has also an advantages to the patient and also to the scientists, into the nature and in our environment. These are: gene therapy has the potential to eliminate and prevent hereditary diseases such as cystic fibrosis, ADA, SCIED, and etc. It is a possible cure for heart diseases, AIDS, and cancer. And nowadays, that this gene therapy is now using for the cancer, for leukemia, and some other cases. Also, gene therapy gives someone born with a genetic disease a chance to life, because some babies are born with hereditary diseases. Gene therapy can be used to eradicate diseases from the future generations. There is also an ethical issues about gene therapy. Number 1. Who will have access to therapy? Is it interfering with God's plan? Should people be allowed to use gene therapy to enhance basic human traits such as height, intelligence, etc.? Is it alright to use the therapy in the prenatal stage of development in babies?
Audio Jungle.